Arabinose Operon. Like lac-operon, Arabinose Operon also regulates at the initiation of transcription. However, unlike lac-operon's simplicity, Arabinose Operon is comparatively much more complex. So in this session, the focus is basically going to be on the structural and the functional aspects of this operon. So let us look at the learning outcomes of this session. So AraBAD has the structural genes that are enabling breakdown of L-arabinose in E. coli. And other than AraBAD are two more clusters, AraE and AraFTH. These two are solely responsible for uptake of arabinose. Now, other than E. coli, Arabinose operon is also present in some other prokaryotes, but there are observed certain structural and functional differences, although the general mechanism is more or less the same. This is an operon that can be regulated by both activation and repression, with the phenomenon of DNA looping playing a very important role in regulation. In fact, the first time that DNA looping was observed to be playing a role in regulation was through the Arabinose operon. AraC is the regulatory protein that regulates depending on whether there is glucose present in the media or there is arabinose present in the media or there are both of them present in the media. It is an operon that has dual positive and negative control, also bringing in a concept of anti-activation and also autoregulation. So let us get an overview of what is the function of the Arabinose operon. So the presence of Arabinose operon in the E. coli enables the E. coli to use arabinose as an alternative source of carbon. And this is primarily done by catalyzing the arabinose to xylulose 5-phosphate. The entire catalysis of this, or the entire pathway, or the entire utilization of arabinose is solely dependent on the three clusters that are present in the arabinose operon. So primarily the pathway is L-arabinose is getting converted to L-ribulose by the enzyme L-arabinose isomerase. And L-ribulose is then converted to L-ribulose 5-phosphate by the enzyme L-ribulokinase. And then L-ribulose 5-phosphate is converted to D-xylulose 5-phosphate. So the enzyme that is able to convert ribulose into xylulose is L-ribulose 5-phosphate epimerase. And then, of course, xylulose 5-phosphate enters the pentose phosphate pathway. Now, the operon, as mentioned, has the ability to take up the arabinose into the cell from the external environment. Only when the arabinose enters the E. coli's cytoplasm can it be converted into xylulose 5-phosphate. Now, therefore, let us now look at the structure function aspects. So, first the construct of the arabinose operon and then relate the structure with the function. So, the Arabinose operon is in fact a long stretch of DNA, as there are three clusters present, and in fact, what has been observed is that these three clusters are not present very close to each other. In fact, they are a little spaced out. So, the three clusters that are there are one is AraBAD, the second is AraFGH, and the third is AraE. So, it is in this order because the distance when you map the Arabinose operon, the closest is the AraBAD and the farthest is the AraE. Now, the AraFGH has three genes F, AraF, AraG, AraH, and of course they are being expressed through a promoter specifically for FGH. Similarly, you have AraE with its own promoter, and it gives rise to an enzyme which is required for utilization of arabinose. Then, of course, you have the AraBAD under the control of promoter BAD, that is PBAD, and these three structural genes are transcribed at one go, so they are basically polycistronic. And of course, in the AraBAD itself, upstream of this P promoter BAD is present another gene which is responsible for giving rise to AraC, that is the regulatory protein, and this is totally under the control of its own promoter, which is PC. Now, the AraE, is responsible for giving rise to arabinose permease. So as the word suggests, arabinose permease is a carrier protein for the arabinose, and therefore it is present in the cell membrane of the E. coli, so that the permease is able to transport the arabinose from the outer environment or from the media in which the E. coli is growing to within the E. coli per se. The FGH give rise to three proteins which help the permease to take up the arabinose. So, due to both arabinose permease and the AraFGH, the arabinose is channelized into the cell. Now, the structural genes, that is BAD, they are not in order when one looks at the pathway per se. So the first enzyme responsible for converting L-arabinose into L-ribulose is the AraA, which gives rise to L-arabinose isomerase. Then the L-ribulose is converted to L-ribulose 5-phosphate by the enzyme L-ribulokinase, which is a product of AraB, and AraD gives rise to L-ribulose 5-phosphate epimerase, which is responsible for the final product, that is D-xylulose 5-phosphate. AraC is the regulatory protein, and this in fact is regulating all the three clusters, not just AraBAD, but it also is able to regulate AraFGH and AraE. Now, what is another interesting facet is that for the promoter for BAD, the RNA pol, when it comes and binds, it's synthesized in the direction that is shown. But the RNA that binds to the promoter of the regulatory protein, that is AraC, that is actually synthesizing the mRNA in the opposite direction to that of the structural genes. So, understanding is therefore that RNA polymerase is always going to synthesize in the 5' to 3' direction. So, the way the RNA pol is orienting itself onto the DNA depends on where or which strand has the -10 and -35. So eventually the RNA pol is always going to bind in such a way that the template strand is getting transcribed. So in this case, therefore, the RNA pol will bind in such a way that it transcribes in this direction, whereas the RNA pol in this will bind in such a way that it transcribes in this direction. So this is something that is interesting to note. Now, we are going to focus, or this session is basically going to focus on the regulation at this cluster, that is the AraBAD cluster, because compared to the AraBAD, in AraFGH and E, the regulation is much simpler. So understanding the regulation at the AraBAD cluster is going to give an idea of how the Arabinose Operon is going to function. So therefore, to focus on the AraBAD cluster, let us look at the organization of the AraBAD complex cluster. So you have the structural genes which are regulated, or sorry, which are expressed through the promoter for specifically BAD. So you have the promoter for BAD. Then just upstream of the promoter for BAD, you have two regions which is called as AraI1 and AraI2. And just upstream of AraI1 and AraI2 is the cap binding site. All of you are familiar with cap binding site because you have the same cap binding site in the lac operon as well. So one other thing that you would notice over here is that cap is not just being the activator for the lac operon, but cap has a role to play in Arabinose operon as well, which means that CAP is carrying out what is called as combinatorial control. In other words, it means that CAP is responsible for acting as a activator for not one operon but several operons. And that is interesting. Why? Because CAP is generally dependent on the amount of glucose present in the media. So if glucose is present in the media, none of the other operons that utilize the other sugars have to really be active. And therefore, it is this just one activator that is controlling the operons when glucose, depending on whether glucose is absent or glucose is present. Now, just upstream of the cap binding sequence is operator region, which is O1L, L stands for left, and O1R. Now, just like in the lac operon, OL1 and OLR are also having two half sites. And please note that the AraI1 and AraI2 also are half sites, but with a difference. Here the half sides have two-fold symmetry. Here the two half sides are not exactly the same. There is a difference, and hence the binding of a molecule to AraI1 and AraI2 may vary depending on what the conformation of the molecule is. So this is followed by upstream the promoter of the regulatory protein, that is AraC, and you have from here the AraC. This is the gene for the AraC, but within the gene for AraC is present another operator region with one half site, not two half sites, but just one half site. And this AraO2, this is what is the operator region within the C region, that is the AraC region. So this plays a very important role in the entire regulation process, which we will understand in some time. Now, in this construct, from the cap binding sequence to AraI2, this region is what is called as the initiator or the activator region. So it is this, it is binding of the regulatory protein to this region that controls or that activates the synthesis of the structural genes. Now, the AraC is responsible for synthesis of the regulatory protein, which is the AraC. And interestingly, let us look at the structure of this in a little more detail. Interestingly, the AraC has two main domains. This is the C-terminal domain and this is the N-terminal domain. The C-terminal domain has a DNA binding domain, and what has been observed is that this too has a helix-turn helix motif, but there are more structural components present apart from the helix-turn helix also that finally decides the conformation of the DNA binding domain. It has a linker that is it is part of the entire AraC itself, but it has a polypeptide which acts as a linker or a hinge between the C-terminal domain and the N-terminal domain. And this part of the N-terminal domain is the dimerization domain. We will look at how the dimerization happens. Plus you have the N-terminal arm over here. Again, you have the connect between the C-terminal domain and the N-terminal domain. And it is this N-terminal domain which also has the arabinose binding site or arabinose binding pocket. So therefore, you can understand that the AraC on one hand is able to bind to DNA and on other hand it is able to bind to arabinose. Now, we talked about N-terminal dimerization. So, what is interesting to note is that When the Arabinose binds to the Arabinose binding pocket, the entire AraC changes its conformation. So in fact, therefore, AraC exists in two conformation. One is this conformation as you see. So this is the conformation you see, and if you have all the AraC present without binding to Arabinose, then at this dimerization point you will have two AraC coming and binding to form a dimer in this structure or in this conformation. But if you have the Arabinose bound at the Arabinose pocket, you can see that due to the hinge region, there is a change in the conformation. The N-terminal domain bends towards the C-terminal domain to form a structure like this. Now, this structure can also dimerize. So, if you have another AraC with Arabinose bound, then you would have basically the dimerization happening like this. So, look at this fact. With Arabinose, the two AraC do dimerize, but look at the conformation that it gets. It is way different from when you have dimerization of two AraC without the Arabinose. So this conformation and this conformation therefore would have a different role to play. So it is a perfect example of how what the structure is decides what the function is going to be. Now, let us look at therefore what happens when you have the E. coli growing in a media that contains glucose, but it does not contain arabinose. So the idea here is that if glucose is present and arabinose is absent, obviously the Arabinose operon should be switched off. So let us look at how it is switched off or what happens that makes it get switched off. So this was the basic construct of the AraBAD. So we will focus only on the AraBAD. Now, when glucose is present, okay, the cap is inactive. This is something that we have learned from the AraC operon itself. Only when you have glucose absent, the cyclic AMP level is going to be high. And when cyclic AMP level is going to be high, it will bind to the cap. On binding to the cap will have affinity to bind to the cap binding sequence. But if glucose is present, then ATP is high and cyclic AMP is going to be low. So you will not have much of the cap having the cyclic AMP bound to it, and therefore the conformation of cap will be such that it is not able to bind to the cap binding sequence. So in other words, what we are trying to look at is cap is an activator, but in presence of glucose, cap is not going to bind. So through cap, you cannot have the AraBAD getting expressed. Okay. Now, the second scenario is that since the promoter for BAD is accessible, RNA pol can come and bind to the promoter for BAD. However, what has been observed is that the promoter for BAD has both its -35 and -10 deviating to quite an extent from the consensus sequence, and therefore you will not have the RNA polymerase carrying out transcription, because although it may bind, the interaction is so weak that it cannot go from a closed complex, or it cannot transit from a closed complex to a open complex, and only when it forms an open complex will it carry out transcription. So therefore, when the interaction of the RNA polymerase with the promoter for BAD is so less, the RNA pol will bind and then leave and bind and leave. So therefore, you will never have a stable interaction at this, and so you would have generally no transcription of the structural BAD. Sometimes it may happen that it does give rise to one or two copies of the BAD by weak binding if it is able to go to open complex and then synthesize it. So you may have very negligible amounts of BAD present, but what has been reported is generally there is hardly any level of BAD present in the E. coli. Although cluster E, okay, the promoter of which is also controlled by AraC, that is able to produce minimal levels of enzyme, and so permease is present in the cell in a certain amount. Now, when glucose is present, okay, the promoter for the AraC is again accessible to the RNA pol, and as you can see, the RNA pol can come and bind to the promoter for C and start synthesizing the AraC. So you can see that over a period of time, you will slowly, gradually have many AraC being formed. And this AraC, in this conformation, when it is not bound to arabinose, because arabinose is not present, it is not going to bind to the arabinose binding pocket. So when the arabinose is not bound, you know that it has a certain conformation, and this conformation, in this conformation, the DNA binding domain has affinity to bind to the operator 2, which is the half site, okay, and it has only one half site. And you can also have ARA I1, okay, this is the half site to which the DNA binding domain of the AraC without arabinose can bind. So now what you observe is that ARA I1 has one monomer and ARA O2 has the other monomer, and the N-terminal domain of the two are free, so therefore dimerization can happen. Okay, now therefore what is going to happen is, if this has to bind to this to dimerize, then the DNA has to loop. And so you would obviously see the DNA looping such that you have the dimer formed, and this dimer holds at one end the ARA O2 and at the other end the ARA I1. Now, when you have this DNA looping, what is happening is that the steric hindrance is increasing. Okay. Moreover, what has been observed is that when the AraC, as it is formed by this RNA pol, when the AraC exceeds a certain number, so if it is that there are more than 40 copies of the AraC already available, then the AraC in this conformation can also go and bind to the operator regions over here. So you have two AraCs coming and binding, as there are two half sites, correct? But they wouldn't dimerize really, but you would have individually the the AraC coming and binding. Now when you have the AraC bound to O1 and O2, and this O1 and O2 is overlapping the promoter for AraC, then what you observe is that in this case again, because you have the steric hindrance, this RNA pol is not able to bind to the promoter for BAD, and this RNA pol is not able to properly bind to the promoter for AraC, and therefore you will not have transcription of the BAD, that is the structural genes, nor would you have a transcription for the AraC possible. So it means that the AraC, after a certain concentration or after a certain number, is able to auto-regulate its own synthesis. When AraC comes and binds to the O1 and O2 in proximity to PC, then the RNA pol is not able to initiate transcription, and so what is going to happen is that the level of AraC will be maintained. If the AraC decreases in number, then these two would leave the O1, O2, and you would have, because it leaves, you would again have the polymerase able to synthesize the AraC. So therefore, this is a very interesting facet of the Arabinose operon, which means that the regulator protein is able to regulate its own synthesis. And this phenomenon is what is called as autoregulation. Now, this that in this conformation, when it is a dimer and in this conformation, then it is acting as a repressor, because the AraC itself is not allowing initiation. So it is anti-initiation. Okay, now let us look at glucose is present, arabinose is also present. So in such a scenario, what is going to happen? So one of the first things that is understandable is, because glucose is present, ATP amount is high. Okay, and since ATP amount is high and cyclic AMP is low, you will not have cyclic AMP binding to CAP, And if cyclic AMP is not bound to cap, it is not going to bind to cap binding sequence. And so one of the activators is not functioning. But see, arabinose is present. So if arabinose is present and you have a certain number of arabinose permease present on the membrane, so arabinose will be taken up by the E. coli, which means that you will find that the arabinose is able to bind to the AraC at the at the arabinose binding pocket. And when that happens, we know that its conformation is going to change. So this conformation is going to change, right? And now you would have, with the change in conformation, the dimer that is formed with the arabinose binding to AraI1 and AraI2. So with the arabinose bind, the DNA binding domain of this is now able to bind to AraI2. If the arabinose is not present, then this will not be able to bind to AraI2. So you can now observe that the entire AraC dimer is bound to AraI1 and AraI2. When these have been bound to by the arabinose, you will find that its affinity to bind to the operator region is less and they would be released from the operator regions. So currently, therefore, you do not have a on the operator region for the promoter for the AraC, and therefore you can have the RNA pol coming and binding over here and transcribing. While in this case, the RNA pol can come and bind to the promoter for BAD. But what has been observed is in the absence of cap binding to the cap binding sequence, again, this is not fully a very strong interaction. And even though there is presence of AraC, which is acting as an activator over here, it still is not able to fully activate the RNA polymerase to carry out transcription. So you will not have transcription over here, and this will transcribe. So now one is not very sure of the exact mechanism or what is exactly happening when both are present, but what is definitely the case is that the Arabinose operon is going to be repressed when glucose is present. Now let us look at the scenario where the glucose has been completely utilized and Arabinose is present. So when Arabinose is present, therefore the requirement in E. coli is that the Arabinose operon has to be switched on. So therefore, when you had glucose present, the Arabinose operon was switched off due to the looping of the DNA and the dimerization of the AraC such that it acts as a repressor. And therefore, you cannot have RNA pol coming and binding to the promoter for the structural genes to carry out synthesis of the structural genes. And hence, if you do not have the structural genes to utilize Arabinose, you will not have Arabinose being utilized. But now the condition is different. You don't have glucose, but you have Arabinose. So therefore, what is the understanding is that with the glucose absent and arabinose present, you will have the AraC being formed. But because arabinose is present, you know that arabinose is going to come and bind to the AraC, and with that there will be a structural conformational change. And because there is a structural conformational change, you will have the dimerization happening in such a way that the DNA binding domain of the AraC will bind to AraI1 and AraI2. So this conformation of the AraC is going to act like an activator. However, what is observed is that the cap, which is now able to come and bind to cap binding site. Why? Because glucose is absent. So the levels of ATP is going to be low and the levels of cyclic AMP is going to be high. So cyclic AMP being high, it will come and bind to the cap, and now the conformation that cap gets, it is able to come and bind to the cap binding sequence. So together, the cyclic AMP cap plus the AraC complex is able to enable the RNA pol to attach or to bind to the promoter for BAD with a very strong interaction. We all know that the RNA pol has what is called as the alpha C-terminal domain tail, the alpha CTD tail. So the alpha CTD tail is both binding to the cap as well as it is binding to AraC. And when both of these bind to the alpha CTD tail, the conformation of the RNA pol is such that it is able to interact with the promoter for BAD strongly and get transited from the closed complex to open complex. And when it forms the open complex, the transcription bubble is formed, and with the transcription bubble forming, you have the synthesis of the mRNA, giving rise to the three enzymes that are required to utilize arabinose. So that is how, when you have the conformation of the AraC such a way that it can bind to the activation region, then it is able to allow transcription to happen. Now, this is a perfect example of cooperative binding. The presence of the activator and its attachment to the alpha CTD tail of the RNA pol is enabling these two to recruit the RNA pol to the promoter region and enable it to carry out transcription. So, an example of cooperative binding. But this is also an example of allostery, because only when the arabinose binds to the AraC, it takes up a conformation that enables it to act as an activator. Otherwise, in this form, it is acting as a repressor. So, what is very, very interesting about Arabinose Operon is that AraC is able to take up two different conformations such that one conformation makes it a repressor and the other conformation makes it an activator. Thus, let us make the concluding remarks. The Arabinose operon consists of three clusters with ARA-FGH and ARA-E responsible for uptake of arabinose and ARA-BAD responsible for its utilization. The ARA-BAD is responsible for converting L-arabinose into D-xylulose 5-phosphate, and like mentioned earlier, this then enters a pathway that allows it to be used as the carbon source. AraC is the regulatory protein of the Arabinose operon, and this AraC has a DNA binding domain as well as it has an arabinose binding pocket. So when AraC is bound to arabinose, it acts as an activator, and when it is not bound to arabinose, it acts as a repressor by enabling what is called as DNA looping. So with DNA looping, there is a steric hindrance, and therefore the RNA pol cannot come and bind to the respective promoters. When AraC exceeds a certain number in the cell, it autoregulates its own synthesis. The cyclic AMP cap complex along with AraC recruits RNA pol to the promoter for BAD and initiates transcription. Therefore, Arabinose operon is a paradigm for a regulatory protein with dual role of positive as well as negative control, because it is the same protein that can act as a activator and it can also act as a repressor. Thereby, when needed, it ensures that there is no initiation happening. Moreover, the regulatory protein is able to autoregulate its own synthesis. Thank you.